Welcome back from the lunch break. We hope you're able to have a nutritious lunch. To get things started, we do have a few housekeeping notes regarding CE information. As a reminder, in addition to the speaker evaluation, we have our final evaluation, which will be available later this afternoon from our one-stop shop link. This final survey is the mechanism by which you will receive the certificate of attendance, which we'll use to support CE credits for RAP, SOCRA, SQA, and ACRP. Be sure to download the certificate immediately upon completing the survey. Otherwise, you will miss the opportunity to do so. Even if you do not want or need the certificate, please take a few minutes to complete the conference evaluation, as your feedback will help guide the design of our future events. Note that the evaluation and certificate are only available for 2 weeks. That takes us to October the 5th. To summarize, for our physicians, pharmacists, and nurses claiming continuing education credits, CEs are only available if you've registered via Adobe Connect and attend in real time. SBIA will issue the CE claim code and instructions to all attendees later today. Be sure to complete the CE evaluation on or before October the 5th. I apologize in advance, but we are unable to make exceptions to that deadline. Attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to their attendance to claim credit. Today's email will have both the claim code and instructions, together with the survey link. As a reminder, all sessions are being recorded. It'll take us a week or so to process the recordings, but you will soon be able to access them from SBI's webpage. And I'm sure you've got that bookmarked, but let me give it to you again just in case. It's fda.gov/CEDAR SBIA Learn. This leads us to session 7 on quantitative methods, study design, model integrated BE approaches. Our first presentation in session 7 is on alternative model-based data analysis approach to demonstrate BE. Our presenter, Dr. Yuqing Gong, is currently a staff fellow at the Quantitative Clinical Pharmacology team in the Division of Quantitative Methods and Modeling, ORS-OGD. Her current role in the division is to utilize quantitative tools such as population pharmacokinetics, modeling, and simulations to address specific questions that relate to generic drug development process and/or regulatory decision making. Our second presentation is on the evaluation and application of new and novel data imputation approaches to support BE assessment. Our presenter, Dr. Jenny Wang, joined the Division of Quantitative Methods and Modeling of ORS-OGD as an ORISE Fellow in October 2020, and she worked on multiple projects, including data imputation, methodology for clinical trials, and incorporating machine learning algorithms to enhance PSG review efficiency and bioequivalence assessment. Our third presentation is on challenges and opportunities on using oral PBPK to support risk assessment and Biowaiver in regulatory submissions. Our presenter, Dr. Feng Wu, is a senior pharmacologist and reviewer and scientific lead for oral physiologically based pharmacokinetic modeling in the Division of Quantitative Methods and Modeling. Dr. Wu has been with FDA for more than 10 years. She's responsible for using modeling and simulations tools for reviewing pre-ANDA meeting packages, ANDA consults, and controlled correspondences. Prior to joining DQMM, Dr. Wu was a biopharmaceutics reviewer for more than 4 years and responsible for NDA and ANDA biopharmaceutics reviews. Our final presentation is on the Dermal PBPK modeling for a transdermal delivery system to assess the impact of the application site on in-vivo performance. Our presenter, Dr. Eleftheria Cyclazal, joined FDA in 2015 as an ORISE Fellow. She is currently a staff fellow at the Division of Quantitative Methods and Modeling. Her interests include dermal physiologically based pharmacokinetic modeling, interactions between excipients and molecular targets, including gut transporters, and development of quantitative modeling and simulation tools to support bioequivalence assessments. Please join me in welcoming our first presenter, Dr. Yuqing Gong.
Thank you for the nice introduction. Here, I'd like to share the use of alternative model-based data analysis approaches to demonstrate bioequivalence. This presentation represents the views and perspectives of the speaker and does not necessarily reflect the views of the US FDA. This talk will help you recognize opportunities of using a model-based approach as an alternative data analysis method to demonstrate bioequivalence. And we will review a case example together of using population pharmacokinetics, PBPK modeling as such an alternative analysis approach. We will also learn what are the key regulatory considerations when an alternative model-based approach is used. What are the key components that should be included? FDA has recognized opportunities of using alternative model-based approach to demonstrate BE in the product-specific guidance of paliperidone palmate extended release suspension for the 3-month injection. And using such a model-based approach for bioequivalence establishment was one of the topics that has been discussed at FY 2022 Generic Drug Science and Research Initiative Workshop. For example, how a broader adoption of modeling could be done to overcome bioequivalence challenges that are otherwise difficult to resolve. As you may know, a wide range of modeling approaches can be implemented into bioequivalence assessment. In this talk, I'm focusing on the use of quantitative clinical pharmacology tools, such as population pharmacokinetics modeling, to address challenges in pharmacokinetics bioequivalence studies, PKBE studies. Here are some examples. Modeling can be used as alternative data analysis approach for an interrupted BE study. For example, for a study interrupted by the pandemic that caused the missing data in the study. In addition, modeling can be used to develop alternative study designs or analysis methods for challenging drug products, such as long-acting injectable products where the conventional BE study is difficult to conduct due to the long study duration. Modeling can also be used for product for rare disease, where the patient recruitment is difficult and may also be restricted by the dosing regimen. Last but not the least, modeling can also help for studies with sparse PK sampling. Here, I'm only listing some examples in the PKBE study. There are more opportunities with different types of studies or products and different types of modeling approaches. Now let's review a case example together that used population PK modeling approach to demonstrate BE for an interrupted study. Here is the potential process that can be used to address an interrupted BE study. First, of course, the applicant will identify the issue before unblinding the data. Then the applicant may propose a solution that could be the alternative analysis for the interrupted study. In order to clarify FDA's expectations early and for FDA to assess the prospective applicant to submit ANDA as complete as possible, the applicant may consider discussing the proposed alternative analysis plan with the FDA via pre-ANDA interactions, such as control corresponds or pre-ANDA meetings. Please note the statistical analysis plan changes should be made prior to data lock and unblinding, and this alternative analysis should be accompanied with adequate scientific justifications and not lead to biased bioequivalence determination. This is a simulated case example for an interrupted PKBE study. This is a single-dose in vivo PKBE study with long study duration. Due to the pandemic, many subjects could not return to the clinic to provide their PK samples. As you can see from the PK profile, the shaded area represents the study duration that was impacted. This study experienced a high volume of missing samples in the mid to late phase. For one time point, it can be as high as 70% missing samplings. As you can see from the graph, there will be subject with interrupted PK profile, as well as subject with truncated PK profile. This consecutive missing sampling in elimination phase impacts AUC calculation when performed the conventional non-compartmental NCA analysis. And we will have interrupted AUC profile, as showed at the left, that a time duration is missing for the data, or the truncated AUC profile, as shown at the right, where the last sampling time point is truncated. In addition, because of the missing samples in the elimination phase, this can cause issues in estimating terminal rate constant lambda, so the AUC infinity may not be able to estimate accurately, or may not even be able to calculate. Here is what modeling can help. PK modeling can be used to impute missing values. The graph here demonstrates how the PK modeling will do here. For this case, data imputation is conducted at an individual level. In this graph, for the observed PK profile for a subject, it's shown in red solid line. The population PK model is developed from this impacted study. For each subject, we will have a model predicted concentration for all the time points, as shown in the blue dashed line. So for each subject, if a time point has observed value available, we will use the observed value. However, if a time point has a missing value, this time point is then filled by values estimated from the PPK model with uncertainty estimated from the population PK model. This is presented as the imputation area, shown as light blue. Here, the advantages is we will predict the missing part for this specific individual. In the meantime, this prediction is based on the model developed based on the whole population that incorporates subjects who had completed PK profile. Thus, you will have a combined profile which contains the observed value as well as the predicted value for the missing part. This is the overall steps for this PPK model-based imputation in this case example. First, we developed a structured model using data from the reference products, with model selection, assessment of covariates. Then, we included formulation-depending parameters to predict different formulations. Second, we performed PPK model validation and verification, including goodness of fit plots, visual predictive checks, reversing the population PK guidance. Third, we imputed data for missed visits for each individual. To account for uncertainty from the residual variability, we conducted 1,000 times imputations and calculated the passing rate from these 1,000 imputations. In the end, PK profile with model imputed data are used for BE establishment, and observed data with conventional NCA approach are used as supportive information. We performed BE for all PK parameters including Cmax, AUCT, and AUC infinity based on PK profile with missing values imputed. And data imputation were conducted for 1,000 times to account for uncertainty from the residual variability. And from this case example, the passing rate from 1,000 imputations was calculated to be 100%. So here, we demonstrated an example case that had high volumes of consecutive missing samples in the elimination phase, where the conventional NCA approach may be insufficient to demonstrate bioequivalence, for example, to have incompleted PK profile. An alternative model-based data analysis approach could help BE demonstration in this case for a pandemic-interrupted study. In this case, a PBPK model developed using the actual clinical data with sufficient validation can be used for data imputation at an individual level to mitigate the impact of missing data points. And there is a reminder that the applicant is expected to determine and pre-specify analysis plan before unblinding the clinical data if they plan to propose such an alternative analysis plan. Here are some key components that is expected to accompany with an alternative model-based analysis approach. There should be adequate scientific justifications, for example, include sufficient model verification and validation for the intended regulatory use, and demonstrate it is capable to distinguish formulation difference and comparable to the conventional approach, and demonstrate it would not lead to biased equivalence determination, and properly characterize the uncertainty and the impact on BE determination. The proposed alternative approach can be discussed with FDA early via the pre-ANDA programs such as control corresponds and pre-ANDA meetings. Alternative model-based approach should be pre-specified in statistical analysis plan and be made prior to data lock and unblinding. Now let's go to challenge question number 1. Which of the following scenarios does not belong to alternative model-based data analysis approaches to demonstrate BE? A. Use population PK model for data imputation for an interrupted BE study. B. Use non-compartmental analysis NCA to calculate PK matrix. C. Use modeling approaches to develop alternative study designs or analysis method for challenging drug products. Take a moment to think about the correct answer. Yes, I think you all got it. The correct answer is B. Use non-compartmental analysis to calculate PK matrix. This is the conventional way to calculate PK matrix and does not belong to alternative model-based data analysis approach we are talking here. Let's go to challenge question number 2. An alternative model-based data analysis approach should A. Be accompanied with adequate scientific justifications. B. Not lead to biased equivalence determinations. C. Be pre-specified before seeing study results. D. All of above. Take a moment to think the correct answer. Yes, the correct answer is D, all of above. All above listed points are considered the key components for an alternative model-based data analysis approach. Here are some resources you may refer to regarding the topics I talked about today. And I'd like to acknowledge all offices that support the effort. For Office of Generic Drug, the Immediate Office, Office of Research and Standards, Office of Bioequivalence, specifically to the following individuals named listed here who get involved directly to this project. Please submit your questions and I will be back in the panel to answer your questions. This is the closing thought. Quantitative method and modeling has been increasingly applied to facilitating generic drug development and review, and play a critical role in the modernization of bioequivalence assessment. Thank you.
Hi everyone, my name is Jing Wang. I work in the Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs, CDER, FDA. Today I will give a presentation about advanced in data imputation approach to support BE assessment, to introduce our current research work on data imputation method. Before my presentation, I would like to disclaim that this presentation only reflects the views of the author and should not be construed to represent FDA's views or policies. This is the outline of my presentation today. I will first introduce our motivation for this research work, and then I will introduce the background, method, and performance test information about this research. At last, I will introduce a hypothetic case study. So in current practice for bioequivalence assessment, the applicant's submitted protocol should include plans to minimize missing data and pre-specify statistical analysis for treating missing data with sufficient justifications. Generally, replacement of missing values using data imputation methods is not recommended. Under some special circumstances, data imputation methods could be potentially used to provide additional evidence to support BE assessment. This presentation reports a recent research study in developing a new data imputation method that could be potentially used to support BE assessment. And this work should not be considered as an FDA recommended method for ANDA submission. Missing data commonly exists for PK or PDB studies. There are different types of missing data. The first type is missing not at random, MNAR. That means the propensity of the values is related to the value itself. For example, the data censored due to the detection limit. The second type, missing completely at random, MCAR, that means the missingness of the data has nothing to do with the data value itself. It may be due to some random events. For example, a blood sample can be damaged in the lab. The third type, missing at random, MAR, that means the missingness has nothing to do with other observed values, but may be affected by some systematic factors. It is classically assumed that all MAR values are also MCAR, and the same imputation methods for MCAR can also be used for MAR missing values. Missing values in real-world datasets are usually a mixture of these types. Missing data can pose a lot of challenges in BE assessment as it can bias the parameter estimations distort sample distribution, and impair statistical power. Imputation is the process of replacing missing data with substituted values. Scientifically sound imputation methods could potentially be used to support BE assessment. There are existing imputation methods developed for different kinds of missing data, not only for PK or PD data. For example, for the MNAR data, there are methods like half of the minimal value, k-nearest neighbor truncation, quantile regression approach for left-censored missing data, and Gibbs sampler-based left-censored missing value imputation approach called GSIM. I will introduce more details about this GSIM method in the following slide. To handle the MCAR or MAR data, there are methods like k-nearest neighbors, misforest, and imputation with SVD. The reason why we are interested in the GSIM method is that after reviewing the relevant literatures about data imputation approaches, we find this newly developed method, GSIM, which was published in 2018, showed the best performance for the data imputation for MNAR data, that is censored data. Like this figure showed here, this figure is from the original GSIM paper. For each subplot, the x-axis represents the imputed values, so y-axis represents the original values, and this blue dots part is the non-missing values, which lies on the diagonal line of each subplot. The right dots part are the imputed values for MNAR missing values. So the distance between the red dots to the diagonal line showed how large the imputation error is. The closer to the diagonal line, the better of the accuracy. So we can see at this right bottom part is the result for GSIM. It shows the much better performance compared to other three methods. And there are also other literature stated that this GSIM method is also has potentially good performance to handle the MCAR data. After examining the details of the original GSIM algorithm, we find there may be two limitations. The first one is it can potentially produce unstable prediction. Like this figure on the left side showed, the x-axis shows the iteration number from 0 to 100. The y-axis showed the values of the prediction for each iteration. The blue line shows the true value for the generated missing value, and the red line shows the imputation limit used to generate the missing value. Original GSIM used the last updated value as the final imputed value, like this dot here. And so the predictions can vary dramatically across different imputation trials. The second limitation is this algorithm may be underperforming for data with mixed MNAR and MCAR values. Original GSIM was developed mainly for MNAR data. It will recommend using a high and low limit setting to constrain the imputed values for MNAR to within a range. Original GSIM also recommend that when dealing with MCAR data, it can use a no limit setting to impute for the MCAR values. Look at the figure on the right side here. The red dot shows the MNAR value, and the green dot shows the MCAR value. The upper part, the x-axis shows the imputed values, and the y-axis shows original values. The diagonal line shows the gold standard for the imputation. The closer the dot is to the diagonal line, that shows a more accurate imputation result. We can see when dealing with the mixture of MNAR and MCAR, if the original GSIM was using the with limit setting, the imputed values for MCAR will be very far away from the diagonal line. And if you use the no limit setting, even though the imputation for MCAR, the green dots are closer to the diagonal line, but it failed to constrain the imputed values for MNAR, the red dots, under the detection limit. We implement two improvements to develop this improved GSIM algorithm. Firstly, we implement probability-based prediction to improve the reliability and accuracy of imputed values. Second one is to integrate different limits for each missing value based on its missing type, to extend this algorithm to mixtures of missing types. So the first improvement of improved GSIM is to implement probability-based prediction. We use the peak value of the estimated probability density of predicted values in all iterations as the imputed value, like this example showed in the figure of the left side. Instead of using the last updated value as the original GSIM, we collected all the predicted values of all 100 iterations and then estimate the probability density of these predicted values. Then we use the peak value here as the final imputed values. So the figure on the left side shows a case example for the imputation results across 7 different imputation processes. The true value of the generated missing value is minus 1.6752. We can see across the 7 different processes, the imputed values of improved GSIM closer to the true value with a much smaller variance compared to the imputed values of the original GSIM. The second improvement of improved GSIM is to extend this algorithm to mixtures of missing types. We add missing type marks for missing values and then set up different limits for each missing value based on missing type mark. During the imputation process, the algorithm can choose the missing type based limits automatically for each missing value. Like this figure showed here, when handling the mixture of MNAR and MCAR, we can see the imputed values of improved GSIM for both types are very close to the diagonal line, and it also successfully constrained the imputed values of MNAR under the detection limit. Here, I will introduce the settings and results of the performance test. So for the simulation part, we use the 160 by 76 dataset without missing values as the original dataset, and this dataset is from the original GSIM paper. Missing values were generated based on missing proportion and the ratio of MANR type in the mixture. We use like 2 parameter alpha and beta to control. Missing type mark were added for each generated missing value. For the performance metrics, we used normalized root mean square error, NRMSE, NRMSE-based sum of ranks, and scatter plots. For both NRMSE and NRMSE-based sum of ranks, lower value represent better imputation accuracy. So through the scatter plots, we can compare the imputed values and the original values. In the first series of performance tests, we compare the improved GSIM and the original GSIM with MNAR data. So we create a series of simulation datasets with the missing proportion from 0.1 to 0.6. Step in 0.1. And for each dataset, we applied the original GSIMP and improved GSIMP 30 times. Like this figure showed here, the x-axis shows the missing proportion of the datasets. The y-axis shows the NRMSE values after the imputation of 30 trials for each dataset. We can see improved GSIMP always showed significant lower imputation error than the original GSIMP. The pair t-test results also showed that the improved GSIMP had significantly lower NRMSE than the original GSIMP across all missing proportions from 0.1 to 0.6. And the p-value is less than 0.001 for all comparisons. For the second series of performance tests, we compared improved GSIMP and other methods with mixture of missing types. Here we use the case example dataset with the generated missing values. The missing proportion is set to 0.2, and among these missing values, 70% of them are MNAR, and the other 30% are MCAR. For each subplot here, we can see the x-axis represents imputed values and the y-axis represents original values. The blue dot represents MCAR, red dots represent the MNAR, and the yellow dots represent the non-missing values. The non-missing values, these red dots, all lie on the diagonal line of each subplot, which represent the gold standard of the imputation. We can see for the method QRILC, TRKNN, Miss Forest, and half of the minimum value, the imputed values for MCAR and the MNAR are mostly away from the diagonal line. Here, the result of the improved GSIMP showed that the imputed values for both MNAR and MCAR are both very close to the diagonal line, and the imputed values for MNAR has been successfully constrained. So through this series of performance tests, we showed that improved GSIMP outperforms the original GSIMP and other methods for MNAR and MCAR mixture dataset. In the performance test 2, we also compare the improved GSIMP and the other methods with a series of simulated datasets with different mixtures of MNAR and MCAR. We mainly used 4 different types of mixture, like this figure listed here. The mixture from MNAR 100%, 70%, all the way to MNAR 0%. So this figure here, the x-axis of each subplot shows the missing proportion. The y-axis shows the NRMSE-based sum of ranks. So the lower the value is, it shows the better imputation accuracy. The red color represents the performance of improved GSIMP. We can see for each figure, improved GSIMP, the sum of rank values is always among the lowest one compared to other methods. So this result showed improved GSIMP outperforms the original GSIMP and other methods in most cases for MNAR, MCAR, or different mixtures of MNAR and MCAR. Now I will introduce the hypothetic application of the improved GSIMP as a case example. This case example is a PDB study from an approved ANDA. Among the 84 subjects in the submitted data, 24 subjects had 44 missing values in PD endpoint due to detection limit, and the missing proportion is about 0.11. without salient deficiency in study design and implementation, given the high percentage of censored values in the study data. FDA's internal analysis adopted a modern likelihood-based modeling approach, that's the M3 model, to perform data imputation for censored values. As a hypothetic application, the improved GSIMP was applied to impute for right censored values to support the BE assessment. The figure in this slide shows the imputation results of improved GSIMP. The x-axis shows the different dose, 0, 90, and 180. The y-axis represents the PD endpoint in log scale. The black color shows the original data points, and the red color shows the imputed values from the improved GSIMP. The circle shape represents the data points for the reference, and the triangle shape represents the data points for test. We can see the imputed values of improved GSIMP are all larger than the observed values because these missing values in this PD study are all right-censored data. They are beyond the limit of imputation. So this figure showed the imputed results of improved GSIMP has been successfully falls in the appropriate range due to the constraint. So with the imputed values by improved GSIMP, the recommended dose scale analysis shows that the calculated 90% confidence interval falls in the acceptance region of BE. Through this assessment, it can enhance the credibility of data and analysis, thus supporting the decision-making process of BE assessments. So our conclusions are: improved GSIMP outperforms the original GSIMP in imputation accuracy for MNAR data. Improved GSIMP outperforms the original GSIMP and other imputation methods for missing data with mixtures of MNAR and MCAR. The superior imputation accuracy and reliable performance of the improved GSIMP showed the potential to facilitate the decision-making process of generic drug development and regulatory assessment. Thank you.
Good afternoon, everyone. The topic of my presentation today is challenges and opportunities when using oral PBPK to support risk assessment and biowaiver in regulatory submissions. This is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. Through this presentation, the participants will understand regulatory questions that PBPK absorption modeling can help answer in generic drug development. The participants can also learn recent case examples on developing PBPK modeling and conducting virtual BE simulations for supporting regulatory decision-making. As you may have known, the regulatory questions that PBPK absorption model can help answer in generic drug development include the topics shown in this slide. They are all related to BE assessment, including evaluating the impact of changes in critical quality attributes, setting dissolution safe space, assessing the risk of formulation mechanism changes, etc. The PBPK absorption modeling can also be used to predict the impact of food and gastric pH change on BE, predict GI local concentration, support waiver of In-Vivo studies, and the BE assessment in specific populations. This figure shows the general PBPK modeling procedure in ANDA submissions. Generally, the model input include in vitro data, for example, dissolution of both test formulation and reference formulation, as well as available clinical data set. Then use IV and oral In-Vivo data to estimate disposition and absorption parameters, After verification, validation of the model, sensitivity analysis could be conducted to identify the key factors that impact the in vivo performance. The model can then be used to predict in vivo PK of different batches. Virtual BE simulations can also be conducted to set dissolution safe space or to simulate B trials between our reference and test product. This table summarizes the recent highlighted cases where PBPK absorption modeling had impacts on regulatory decision-making. For example, PBPK was used to evaluate the impact of drug degradation at pH 1.2 on BE for BCS-based Biowaiver case. PBPK absorption model and IVIVC were also used to evaluate the impact of non-comparable dissolution profiles of the test and ROD products for lower strength in multi-pH media on their in vivo performance. PBPK absorption modeling and simulation was also used to evaluate the impact of food on BE based on in vivo fasted and a pilot fed BE study and to conduct virtual BE simulations with other study design. In the next couple of slides, I will show a few examples of using PBPK absorption model in the regulatory work. This case example shown here is about an oral tablet product for which drug degradation was observed in the in vitro dissolution studies at pH 1.2. The applicant submitted a PBPK model to address whether the observed drug degradation would impact the drug plasma time concentration profile as well as BE under fasted and fed conditions. After review, we identified some deficiencies with the submitted model. For example, the applicant used the in vitro dissolution rather than solubility profiles to calculate drug degradation rate, which is not appropriate. There could be other formulation factors and/or dissolution conditions that led to slower drug release at pH 1.2. Also, the model developed by the applicant could not replicate the findings from in vivo bioavailability study that evaluated the impact of gastric pH change. On the other hand, the agency further developed a PBPK model showed that the observed in vitro drug degradation is not expected to significantly impact the plasma time concentration profile and drug exposure under fasted and fed conditions using virtual BE simulations. This is likely due to the long degradation half-life, which is more than 10 hours, as compared to the relatively short gastric transit time, which is about 15 minutes. This case example is about using IVIVC PBPK absorption modeling to assess the impact of non-comparable dissolution profiles on in vivo performance. For drug XER tablets, non-comparable dissolution of the test and ROD products in QC media and multi-pH media for lower strength were observed. IVIVC and PBPK mechanistic absorption modeling were used for predicting the impact of non-comparable dissolution profiles of lower strength on the in vivo performance. After review, major limitations were identified on the IVIVC model. For example, the applicant's IVIVC model was not established and validated using formulations with different release rates. The applicant did not use PK data from intravenous or immediate release oral formulations as reference to predict dissolution parameters. The applicant only used dissolution testing data conducted in quality control media for IVIVC analysis, which is not sufficient to address the non-comparable dissolution profiles of the test and ROD products for lower strength in multimedia at pH 1.2, 4.5, and 6.8 buffers. Case example 3 shown here is about using PBPK absorption modeling to evaluate food impact on BE for drug-wide oral tablets, which include active pharmaceutical ingredients with amorphous solid dispersion form. A mechanistic absorption model was developed based on literature data and results from pilot BE studies and used for predicting the BE under fed conditions. We evaluated the risk and the complexity of the formulation of the proposed product and identified the major concerns limitations of the developed PBPK model. For example, as this is a high-risk product, there is a lack of supporting information related to formulation design, manufacturing process, API characteristics, excipients, and other quality attributes of the drug product that may significantly impact the in vivo dissolution and the bioavailability of the drug. Also, the model validation step is based on BE studies, which have demonstrated BE among the tested batches. So challenging the model with in vitro and in vivo data, which show the lack of BE and or using batches with different release rate to support the robustness of the established PBPK model is recommended. So based on recent regulatory case examples and research experiences, here summarize the challenges and also opportunities for addressing these issues. For example, consistent and adequate approach are needed to generate and incorporate biorelevant, biopredictive solubility, biopredict dissolution profiles obtained through either QC or biorelevant method, and the permeability as model inputs. Use all available datasets to validate the model for its intended purpose. Also use dataset from IV or oral solution data with complete absorption and appropriate approaches to estimate disposition parameters. In addition, the model needs to be challenged with in vitro and in vivo data which show the lack of BE and or batches with different release rate to support robustness of the established PBPK model. Also, formulation variations included in model verifications need to be wide enough to avoid extrapolation outside of the tested space of the formulation variations. To show the robustness of the established model, consider assessing type 1 error for BE assessment when using modeling. For example, large number of virtual BE studies may be conducted with appropriate number of subjects using a series of fold changes between your test product and RLD and report the virtual BE results and the BE passing rate. To resolve the challenges and fill the knowledge gap, OJD has conducted research in this area. Here, I would like to provide some updates on the research for supporting the expansion of BCS Class 3 Biowaiver, including one GDUFA-funded contract project, expanding BCS Class 3 waivers for generic drugs to non-Q1, Q2 formulations, and one internal research project, potential utility of PBPK modeling as an alternative BE approach to support biowaiver of non-Q1, Q2 BCS Class 3 drugs. The contract project is to assess the impact of excipients on the dissolution and the permeation of BCS Class 3 model drugs in solid oral dosage forms. The research results may increase the confidence of using certain amount of some excipients for BCS Class 3 drugs. The internal project is to use PBPK modeling to investigate the impact of permeation change on bioequivalence. Regarding BCS-based biowaiver, currently there is one general guidance, ICH-IM9 guideline for BCS-based biowaivers, which was issued in May 2021. This guideline describes that for BCS Class 3 drug products, biowaiver should be supported by the drug substance is highly soluble, the drug product is very rapidly dissolving, all of the excipients between test and reference product should be qualitatively Q1 the same and quantitatively Q2 similar. As you know, biowaiver option is also available for BCS Class 3 generic drugs. For example, the product-specific guidance for hydroxychloroquine sulfate oral tablet also includes BCS Class 3 based biowaiver option. The language regarding the requirement is similar to what is described in the previously mentioned ICH-M9 general guideline. That is, the test product formulation should be Q1 the same and Q2 very similar, and there are appropriate documentation regarding high solubility and very rapid dissolution. Here, I would like to provide some results based on the GDUFA-funded contract project, expanding BCS Class 3 waivers for generic drugs to non-Q1, Q2 formulations. In this project, 5 model drugs were used, including 4 BCS Class 3 drugs. Acyclovir, cimetidine, ranitidine, and atenolol. Minoxidil, a BCS Class 1 drug, was used as a control. An in vitro dissolution absorption system was used to evaluate the permeation of the pre-dissolved model drugs in the absence and presence of 15 commonly used excipients. This table listed 15 excipients that are commonly used in BCS Class 3 drug products, including povidone key 30, HPMC with 2 viscosities, sodium lauryl sulfate, SLS, PEG 400, etc. 3 concentrations, low, medium, and high, were tested. In general, the high test concentration is equal to the highest amount of a given excipient in immediate-release solid oral dosage form according to the FDA inactive ingredients database, and is dissolved in 250 ml. The medium concentration is generally 25% of the high concentration, and the low concentration is generally 25% of the medium concentration. This is a summary of the research results at this stage. 7 excipients, HPMC with 2 viscosities, microcrystalline cellulose, cross-camelose sodium, talc, mannitol, silicon dioxide had no effect on the permeation of any of the model drugs. Another 6 excipients, povidone K30, magnesium stearate, lactose, calcium phosphate, pragelatinized starch, PEG 400, had some effect, either an increase or a decrease in permeation on only 1 or 2 of the model drugs. One excipient, sorbitol, was inconsistent because it affects the permeation of all of the model drugs, but effects were in different directions in 2 separate experiments. The only excipient that produced dose-dependent effects on the permeation of all model drugs was SLS. In summary, based on the research results, most of the excipients tested had little or no effect on the permeation of BCS Class 3 drugs. This project suggests expanding biowaivers to non-Q1, Q2 formulations with a certain range for BCS Class 3 drug may be possible. In combination with these results, PBPK models may be used to assess the impact of excipients on BE. Therefore, as a proof of concept, we have utilized an oral PBPK model of acyclovir immediate release tablet for assessing the impact of excipients and food intake on the BE of generic acyclovir IR tablets using healthy subjects and virtual bioequivalence trials. Parameter sensitivity analysis and virtual BE using PBPK models were performed to examine the potential impact of apparent permeability on PK and BE of this BCS Class 3 drug. As shown in the right figure, with the increase of apparent permeability, AUC and Cmax of acyclovir increased. The virtual BE simulation results suggested that more than 30% change of apparent permeability value for test product due to presence of certain excipient may result in failed BE of acyclovir 800 IR tablet under both fasted and fat conditions. This demonstrates the potential utility of PBPK modeling to assess the impact of excipients on BE. In addition to the contract project that was mentioned earlier, OGD has funded a couple of contracts and grants related to PBPK modeling as listed here. For example, in the year of 2021, a grant project development and validation of a best practice framework for PBPK analysis for biopharmaceutical applications in support of model-informed biowaivers of fat state BE studies for BCS Class 2 drugs was funded to University of Florida, and a contract project, disintegration and dissolution of solid dosage forms and influence of food-induced viscosity on its kinetics, tools, and methodology for bioequivalence and substitutability evaluation was funded to join Gutenberg University. Both projects are related to the evaluation of food impact on bioequivalence. As demonstrated above, we have conducted research related to oral PBPK modeling through both internal and external projects. Here lists a couple of recent publications based on these research projects. You may refer to these publications for details. In conclusion, the cases introduced in this presentation demonstrated that the PBPK absorption modeling and simulation has been increasingly used in generic drug applications, and GDUFA-funded research projects support to fill the knowledge gap Recent oral PBPK impacts on regulatory decision-making include evaluate the impact of drug degradation at pH 1.2 on BE, conduct risk assessment on the impact of non-comparable dissolution profiles of the test and ROD products on in-vivo performance, conduct a risk assessment on impact of food on bioequivalence, and assess a BE study with another study design. As always, FDA also encouraged the sponsors to submit alternative BE proposals with modeling and simulation data. Communicating with the agency at the early stage where controlled correspondences or pre-ANDA meetings is encouraged. Now, through this presentation, we have learned recent applications of PBPK absorption modeling for oral products. Here, I have two challenge questions for the participants to think about answer. First question, for BCS Class 3 based Biowaiver, which of the following statements is not true? A, the drug substance is highly soluble. B, the drug product is very rapidly dissolving. C, all of the excipients should be qualitatively the same and quantitatively similar. D, there is no possibility of expanding biowaivers to non-Q1, Q2 formulations within a certain range. The answer is D. There is no possibility of expanding biowaivers to non-Q1, Q2 formulations within a certain range. This is not true. Second challenge question. PBPK absorption modeling can be used for A, evaluating the impact of drug degradation at pH 1.2 on BE. B, risk assessment of the impact of deviation of dissolution profiles on BE. C, risk assessment of bioinequivalence attributable to food intake. D, all of the above. The answer is D, all of the above. With that, I would like to thank all the people listed here for their support and hard work, especially the reviewers and the scientific assessors involved in the regulatory work. Also, special thanks to those authors who are involved in the publications that I mentioned in the slides. This concludes my presentation, and I will be back later to answer questions during the panel discussion. Thank you for your attention.
Thank you for the very nice introduction. Hello everyone, my name is Eleftheria Tsakalozu. I am a pharmacologist at the Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs. I'm really excited to be here today to discuss a case of dermal PBPK modeling used to assess the impact of the application site on the in vivo performance of a transdermal delivery system. My disclaimer, this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. Learning objectives for today include a discussion on the general workflow in building and validating a dermal physiologically based pharmacokinetic model for a transdermal delivery system. Describe the application of the dermal model in predicting absorption of an active pharmaceutical ingredient through the skin at an application site other than the one the model was validated for. And describe the application of the developed model in predicting the amount of the API remaining in the transdermal delivery system at the end of the wear period. I will begin this presentation with a little bit of background on transdermal delivery systems and in silico methodologies. The focus of this presentation is a dermal physiologically based pharmacokinetic model, so I will give you a little bit of information on what are these type of models. I will then focus on the case study of interest, giving you a little bit of background, what was the question that we are trying to answer, and how the dermal PBPK model that was developed was applied. What was the considerations in terms of development and validation, and I will end this presentation with take-home messages. Transdermal delivery systems are dosage forms designed to deliver the active ingredient across the skin and into the systemic circulation. Matrix-type TDSs, such as the one shown here on the top, involve active ingredients being dissolved or partially suspended in a mixture of adhesives, penetration enhancers, softeners, and preservatives. In the case of a liquid or a gel reservoir type transdermal delivery system, shown here at the bottom, the components are in liquid or semi-liquid form. They are found in a heat-sealed area and trapped between the packing membrane and microporous membrane. Modeling skin bioavailability, in other words, predicting absorption through the skin, can be a challenging process. The anatomy and physiology of the skin is extremely complex, as you can appreciate from the schematic on the right side of the slide. The skin's role is to protect our body from environmental factors. So when a xenobiotic or an active pharmaceutical ingredient is applied on the surface of the skin, the first barrier it will have to cross is the stratum corneum. After the API, the active pharmaceutical ingredient, can cross the stratum corneum, it will permeate through the different skin layers, the valloepidermis, the dermis, and the hypodermis. Absorption through the skin can also take place via the different skin appendages, hair follicles, as well as sweat glands. When the API reaches skin layers such as the dermis that are vascularized, it has the potential to partition into the systemic circulation. So mathematical models that describe how an active pharmaceutical ingredient can permeate through the skin will need to quantitatively describe these different processes. Partitioning from the dosage form into the stratum corneum and then from one skin layer into the next, as well as diffusion within those different layers, are very important processes that need to be taken into consideration. In certain cases, as the API is permeating through the skin, it is also being metabolized. So mass loss should also be taken into consideration. So a skin absorption model, such as the one seen on the left, is typically coupled with a mechanistic PBPK model, a full-body PBPK model, which uses differential equations to describe how the active pharmaceutical ingredient is taken up and then leaves the different organs of the body: the lung, the brain, the heart, the muscle, the kidney, the liver, the adipose tissue. The power of these mechanistic PBPK models is that they can integrate physicochemical properties of the API, formulation attributes, as well as human or animal physiology information, and the variability around these parameters, and predict what would be the disposition of the API in a biological system. At this point, we have established a general understanding of what transdermal delivery systems are, and have also discussed how we can use dermal PBPK models to describe absorption through the skin for APIs that are applied on the surface of the skin as semi-solid dosage forms or transdermal delivery systems. So let's move on the background related to the case study we will be discussing today. The reference listed drug is the Ortho-Evra patch, which contains ethinyl estradiol and norepinephrine. It is a combination hormonal contraceptive indicated for the prevention of pregnancy in women who elect to use a transdermal patch. For the drug label, patients are advised to apply a new patch to the upper outer arm, abdomen, buttock, or back each week for 3 consecutive weeks. Week 4 is patch-free. Per the Orange Book, the RLD has been discontinued for reasons other than safety or efficacy, and the Zulan patch is designated as the reference standard. The Office of Generic Drugs has published a product-specific guidance on ethinyl estradiol norepinephrine transdermal delivery system. The product-specific guidance includes 3 studies. The first one is a bioequivalent study with pharmacokinetic endpoints in healthy, non-pregnant, non-lactating females who are candidates for hormonal contraception. According to the product-specific guidance, the TDS should be applied to the same anatomical site on all subjects, that anatomical site selected from among those recommended for dosing in the approved labeling for the reference product. As such, there are no specific recommendations on the anatomical site that should be selected for this bioequivalent study with pharmacokinetic endpoints. In the case study that I will be discussing today, a generic TDS, the test product, was developed with API loaded amount that was lower than the respective API amount in the reference standard. The applicant established bioequivalence between the reference standard and their test product following TDS application to the abdomen, which is in agreement with the current product-specific guidance recommendations. As you can see on the graph on the right, there's literature showing that there is no impact of the application site on ethinyl estradiol systemic exposure following application of the RLD product. And that brings us to our question. What is the impact of the application site on the API amount delivered into the systemic circulation over the TDS application period, and can it result in dose depletion when the API loaded amount differs between the reference standard and the test product? The agency internally used modeling and simulation approaches to assess the potential for the need for conducting an in vivo BE study with PK endpoints in female healthy volunteers with BAC as the application site. As we developed our dermal PBPK model and our methodology, we made several assumptions. We assumed that the TDS releases the API at the same rate regardless of the application site, which is consistent with the current knowledge of TDS function. Any differences in PK exposure following application at different application site would be the result of differences in skin physiology between those application sites. Other parameters contributing to variability in addition to the application site include number of subjects, study-to-study differences in skin physiology, drug distribution and elimination, and study conduct, how the TDS was applied and conditions under which the TDS was applied. The potential impact of adhesion on drug product performance was considered to be beyond the scope of this work and was not accounted for here. We followed a stepwise approach in developing and validating the dermal PBPK model that was used here. This slide summarizes our workflow. Our first step was to develop a systemic disposition model for ethinyl estradiol. And the second step was to be able to describe the kinetics, the release kinetics of ethinyl estradiol from the TDS. These two components were combined in a dermal PBPK model for ethinyl estradiol transdermal delivery system. The modeling platform that we used for this modeling work did not include the abdomen as an application site. So we were tasked with developing this application site, and I have a slide dedicated to this work that follows up. The next step was to validate our model extensively. We also leveraged observed data to refine certain parameters, model parameters that I will allude to further. And once we concluded that the model was sufficiently verified for its intended purpose, we applied the model in answering our question. Now I would like to provide a little bit more detail on the different steps that I went over in the previous slide. The first step was to develop a systemic disposition model for intravenously administered ethinyl estradiol. Physicochemical properties of ethinyl estradiol as well as PK characteristics were used for the development of this model. Its performance was assessed against literature data as well as in-house data. Permeation through the skin was described using the MPML McDermott model in the SimSip simulator. QSAR models described diffusion and partition through the skin. We used systemic PK data to optimize two parameters, the diffusion coefficient in the valve epidermis and the dermis, in order to improve the performance of the model. And validation of that model was performed using, again, literature data and in-house data. The release of ethinyl estradiol from the TDS was empirically described by deconvoluting systemic PK data. The abdomen was not available as an application site in the modeling platform that we used. So we developed and validated the abdomen as an application site, leveraging data from the RLD as well as literature studies. We use literature to inform the skin surface pH of the stratum corneum in the abdomen, the thickness of the valve epidermis, the dermis, the subcutis, and the muscle in that application site. And we leverage data from the literature to assess the performance of this abdomen model. And as you can see in the panel on the left side of the slide, the predictions of the model, being the black solid line, describe the observed data for different studies relatively well. The next step was to assess the performance of the model, which entails comparing model predictions with observed data. We extracted the observed data from the literature as well as in-house data for validation. And the outcomes of this validation process are summarized here. Panels A and B show the predictions of the model following application on the back and the abdomen against observed data. Panel C shows model predictions following 3 continuous applications for every 7 days per the drug label. And panel D is a dose proportionality study that showed that the model described well the impact of the application area on exposure to ethinyl estradiol. The validated dermal PBPK model for the reference listed drug was used to develop dermal PBPK models for the reference standard and the test product. These models were used to predict API exposure following application of the test product on the abdomen and assess the risk for dose depletion at the end of the application period on the back. By deconvoluting systemic PK data for the test product, as shown on the panel on the left, ethinyl estradiol release rate was described by the model. The validated PBPK model for the test on the abdomen was then used to predict ethinyl estradiol exposure following application of the TDS on the back of female virtual subjects, as shown on the panel on the right. The validated dermal PBPK models for the test and the reference standard were utilized to predict ethinyl estradiol exposure following the application of the patch on the back and the abdomen of virtual female healthy volunteers. Based on our model predictions, test and reference are expected to be bioequivalent, leveraging the developed models within the scope of a virtual bioequivalence assessment. Additionally, no ethinyl estradiol depletion is expected for the test product when the product is applied on the back of healthy volunteers under the labeled use conditions. This case study highlights how a validated dermal PBPK model can be used to predict the API exposure following TDS application to an anatomical site other than the one evaluated in the in vivo B study with PK endpoints, and to assess the potential for API depletion during the application period. Modeling and simulation approaches with a virtual bioequivalence assessment component can be used to support product development and approval for transdermal and dermatological drug products. Their main advantage is that they provide insight on the effect of important formulation attributes that may influence skin permeation of the API. The agency encourages modeling and simulation approaches that are coupled with the virtual bioequivalence assessment when they support an abbreviated new drug application. The agency encourages an interaction between the industry and regulatory agencies that can be initiated through the pre-NDA meeting request program. Since 2014, a number of collaborative agreements that support the development of in silico tools that describe skin absorption have been funded under the Generic Drug User Fee Amendments Program. For your reference, they are listed here. For the modeling work presented here, I would like to thank Dr. Babiskin, Dr. Fang, Dr. Zhao, Dr. Ghosh, and Dr. Rani. For their continuing support and guidance, I would like to thank Dr. Alam, Dr. Walenga, Dr. Tan, Dr. Luke, Dr. Leinberger, and Dr. Zhang. I will be very happy to answer any questions you might have during the panel discussion session. At this point, I would like to thank you for your attention. Our challenge question for today: what are the key elements of the model development and validation workflow presented here? Systemic disposition model for ethinyl estradiol, in vivo ethinyl estradiol release kinetics from the TDS, dermal PBPK model for ethinyl estradiol TDS, model validation and refinement, all of the above. And the answer is all of the above. Thank you.
Thanks to everyone for great presentations. We'll now enter into our Q&A panel. If you haven't had a chance to submit your question in the Q&A chat pod, please do so now. We'll answer as many questions as time allows. I'd like to welcome to our Q&A panel our session lead, Dr. Zhen Zhang, who's a pharmacologist and bioequivalence reviewer at the Division of Bioequivalence 1, Office of Bioequivalence, OGD. He's a subject matter expert for data analysis and conducts research to address complex bioequivalence-related issues, and is involved in working groups to facilitate the generic drug review process. Please also welcome to the panel Dr. Stella Grosser, who's the Director, Division of Biometrics 8, in the Office of Biostatistics in CEDAR. This division provides statistical support to OGD. She's been at FDA for 21 years, beginning as a statistical reviewer for new drug products and serving as a team lead before assuming her current position. Looks like we have some questions coming in right now, and the first few questions will be addressed to Dr. Yuqing Gong, and here's the first question. Is it an acceptable approach to FDA if an applicant plans a parallel single-dose study with truncated design for LAIs, which are appropriate to dose in?
In general, to allow a truncated AUC, sampling should ensure that complete drug absorption phase is covered and characterized. Also, truncation of AUC may not be appropriate for drugs with flip-flop kinetics. Currently, FDA does not recommend truncated design for LAI product, but if you plan to propose such alternative study design and PK matrix for LAI products, this alternative approach needs to be communicated with FDA with sufficient justification. Thank you.
Thank you for responding to that question. We do have another question for Dr. Gong. And here is the question. The alternative model approach should be specified in the statistical plan. To what extent should the plan be detailed for possible reasons for the use of this modeling approach?
So you can propose an alternative approach if it can be scientifically justified. We focus not only on the reason for you to propose such an approach, but more on the scientific justifications. It's expected that you can convince us that the proposed alternative model integrated approach is sensitive to detect formulation difference with confidence and reasonable passing rate and type 1 control. For example, you can perform some modeling and simulation for the specific product you proposed and with your proposed modeling integrated approach and submit some preliminary response to the FDA to discuss how your method perform. In order to clarify the FDA's expectation early and to assist you to submit the ANDA as complete as possible, you can consider discussing your alternative model integrated approach with the FDA via pre-ANDA meetings. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've received some questions for Dr. Jing Wang. And here's the first question for Dr. Wang. How broad do you see the usage of this GSIMP algorithm for data prediction and data analysis for evaluating the bioequivalence of drug products? And do you see it expediting bioequivalence analysis by the agency?
Thank you very much for the question. I think the improved GSIMP method can be used in all kinds of data-related projects, including the data prediction and the data analysis for evaluating different kinds of bioequivalence of drug products, like the in vitro or IVIVC studies. Regarding the agency use, this method and this improved GSIMP method does have the potential to be used in the agency to support the BE assessment, but it should be a case-by-case situation. In current practice for BE assessment, the applicant submitted protocol should include plans to minimize missing data and pre-specify statistical analysis for treating missing data with sufficient justifications. Firms need to provide scientific justifications that the data imputation process would not to bias the results to convince the agency to trust the result. Thank you.
Thank you for responding to that question. We have another question for Dr. Wang, and here's the question. At what stage would FDA consider imputed value to be unreasonable and call the bio study unacceptable?
Thank you very much for the question. There is no set standard for the agency to consider imputed values to be unreasonable, and it is more of a review issue based on the totality of the evidence. Without scientific justification not to bias the results, it would be very challenging to convince the agency to trust the result. Thank you.
Thank you for responding to those questions. Moving on to our next panelist. We've got a few questions that came in for Dr. Feng Wu, and here's the first question for Dr. Wu. Please comment on the role of the biopharmaceutical drug disposition classification system in evaluating biopharmaceutical characteristics of drugs.
Thank you for the question. So BDDCS, this is a biopharmaceutical drug disposition classification system. Consider the metabolism as well. This may be helpful for predicting the bioavailability and predict DDI. For BE study, we are more focusing on the rate and extent of absorption. Currently, we have BCS-based waiver option in the PSG.
but not yet BDCS. Thank you.
Thank you for responding to that question. We do have another question for Dr. Wu, and here is the question. Please comment on considering Biowaiver for BCS2 drugs with less solubility and acidic pH from 1.2 to 2.
Thank you for the question. For BCS class 2 drug, if the solubility is low, so the excipient manufacturing process may significantly impact the solution and absorption. So if you would like to ask for waiver for BCS2 drugs, it would be more challenging compared to BCS1 and 3 drugs. You may consider providing data, for example, excipient manufacturing process comparison comparable, dissolution risk-based B assessment using, for example, using modeling, PBPK modeling. We will take the totality of the evidence, but if you would like to propose that, please using the control correspondence or pre-AND, consider the early discussion venue with the FDA. Thank you.
Thank you for responding to those questions. Moving on to our next panelist. Got a few questions that did come in for Dr. Eleftheria Sakalizou, and here is the first question for Dr. Sakalizou. What specific attributes of the TDS was used as input parameters in the model for prediction of PK?
Thanks for the question. For the development of the dermal PBPK models for the test, the RLD, as well as the reference standard, we used information on the surface area of the TDS, the API loaded dose, and also the model requires as an input the API release profile. There was also an option for predicting that release kinetics of the API from the TDS into the skin using mechanistic approaches, but these did not perform very well, at least for this specific case. What I did not include in the presentation is considering the fact that we deconvoluted the release kinetics of ethinyl estradiol from the TDS, from the PK data. We assessed the performance of the model locally using IVPT data. So this is another level of validation that we performed to increase our confidence in the predictions of the model. Thank you very much for the question.
Thank you for answering that question. We do have another question for Dr. Sakalizou, and here is the question. What are the considerations for validating a dermal PBPK model, and what type of data may be considered for model validation?
Thank you for this question. As I'm sure you have seen in the case study that I have presented here, model validation is a resource-intense process, and it is extremely challenging for specifically for locally acting drug products for which systemic exposure data may not be available, with the exceptions of transdermal delivery systems, of course, and also quantifying drug amounts at the application site or the site of action, which would be the skin, may not be feasible. So for these products, what we are considering is a two-stage approach, a two-step approach, where we're validating the model that is developed for the drug product of interest, and also validating the modeling platform where the model is developed. And there are a number of publications from our division going into detail on these two specific aspects of model validation. Now, to the second part of the question, it is ideal if model validation is performed against local bioavailability data. This can be dermal microdialysis data, biopsy data, or in vitro permeation testing data. It is also desirable to assess the performance of the model against systemic PK data, but this type of data is available mostly for transdermal delivery systems, as we saw here. And less for topical dermatological drug products. Ultimately, we're looking for a model validation process that results into a fit-for-purpose model. And in recognizing all these challenges, we do encourage the applicants to consider interacting with the agency, with us earlier. We can provide input on their on technical aspects of their model and discuss and provide feedback and discuss what are additional considerations for their modeling approach in regards to their product. Thank you.
Thank you for answering that question. Moving on to our next panelist, we have a question that came in for Dr. Zhen Zhang, and here is the question. What do you consider as a good ANDA submission package for quantitative methods in general?
Thanks for your question. FDA published the guidances on the submission format and content for PBPK and population PK modeling and simulation. You may refer to this guidance to prepare your ANDA submission package for modeling and simulation section. If you conduct modeling and simulation other than PBPK and POP-PK, you may use the same rationale to prepare your ANDA submission package. That is, your submission should contain sufficient information and justification for the agency to replicate and evaluate your modeling and simulation results. Thank you.
Thank you for responding to that question. Moving up to the beginning of our panel, we have a few more questions for Dr. Yuqing Gong. And here's the first question for Dr. Gong. Can any other modeling approach besides PBK modeling be used as an alternative analysis approach to demonstrate BE?
Thank you for the question. We do not limit to population pharmacokinetics PPK modeling to be used as an alternative analysis approach to demonstrate BE. You can propose the most appropriate modeling approach for intended use. Thank you.
Thank you for responding to that question. We do have another question that came in for Dr. Gong, and here is the question. What particular advantages does the model integrated approach provide for studies with large missing values?
Thank you for this question. As I mentioned in the talk, here we are talking for the BE study with large amount of missing data, where a conventional NCA method cannot calculate AUC accurately. So for the presented case, when predict the data for the missing value, it is using the model that is developed based on the whole population that incorporates subjects who had the completed PK profile. So the model will be able to capture the overall PK shape based on the population PK profile and impute the missing value for each subject. Thank you.
Thank you for responding to that question also. Moving on to our next panelist. Have a few more questions that just came in for Dr. Jing Wang. And here's the first question for Dr. Wang. What are the advantages of the improved GSIMP method compared to the conventional missing data replacement methods?
Thank you very much for the question. As we show the performance test results through the simulations in the presentation, that this improved GSIMP method showed much better imputation accuracy than the conventional missing data replacement methods, and it can best preserve the original data distribution. So that's the main advantages. Thank you.
Thank you for responding to that question. We do have another question for Dr. Wong, and here is the question. To handle mixtures of different types of missing data, what data pre-processing procedures may be needed, and how may we define the constraints for each type of missing data?
Thank you very much for the question. To handle the mixtures of different types of missing data, one step in the data pre-processing procedure will be needed is to identify the type of the missing data. Use your best knowledge. For example, based on the situation for each missing data, you may need to give a type number based on the best knowledge. For example, like number 1 for the missing not at random data, like the sensor data, and number 2 for the missing completely at random data. To set the constraints for each type of missing data, for the NAR data, it depends on the, like if it's left sensor data, you may set the constraint to 0 to the lowest limit of the quantification. If it is right sensor data, you can set the constraint to the highest limit of the quantification to infinity, or a reasonable number based on the content. For the MCAR missing data, it's also based on the content of the outcome. You can set a reasonable range for the constraints. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we have another question, a few questions for Dr. Fang Wu, and this is rather lengthy, so here is the question. For a Class 3 API containing drug products, the excipients that might affect bioavailability like sorbitol, mannitol, sodium lauryl sulfate, or other surfactants, do we need to conduct separate studies to show their possible impact on gastrointestinal motility, susceptibility to interactions with drug substances, drug permeability, and interaction with membrane transporters, or are literature searches and citations sufficient?
Thank you for the question. So as I mentioned in my presentation, we have a contract project. The aim actually is to identify a list of low-risk excipients. For those low-risk excipients, if they are used in the generic drugs, and that may be used to support the non-Q1, Q2 Biowaiver for PCS class 3 drugs. What you mentioned here, so they are like SLS, they may consider as high-risk excipients. So you may consider conduct the In-Vivo study, or PBPK can be also used for the risk-based assessment, help for supporting your aim. Thank you.
Thank you for responding to that question. We do have another question that came in for Dr. Wu, and here is the question. As one of the challenges of incorporating biopredictive dissolution and permeation data into PBPK models, could you please describe how the biopredictability is established for dissolution and permeability data?
Thank you for the question. To establish predictability regarding dissolution method, you may use a QC media as a starting point. Then use biorelevant media for generating dissolution data. For some cases, although biorelevant media were not used for generating dissolution profiles, but biopredictability can still be established. Also, to establish biopredictive dissolution method, we recommend you mimicking the In-Vivo situation. And for example, for pediatrics, smaller volume may be used for generating biopredictive dissolution. For predicting fat condition, biorelevant media with increased viscosity may be used. For permeation testing, as mentioned in the presentation, we have conducted research investigation, which will help establish an in vitro permeation method that can be reflective of in vivo situation.
Thank you for responding to that question. Moving on to the next panelist. Got a couple of questions for Dr. Eleftheria Sakalazou. And here is the question for Dr. Sakalazou. How is the agency applying M&S approaches in the case of dermatological drug products?
Thank you for the question. This is a great question and a great opportunity to highlight the constantly increasing confidence of the agency in these in silico methodologies that can be used to support decision making. As we saw in the case study that I presented today, the agency modeling and simulation approaches are employed to examine what-if scenarios. In this case, the question was, what would be the exposure if the TDS was applied on the back instead of the abdomen? Modeling and simulation approaches are also used to assess the impact of deviations in the composition of the test product when compared to the reference. In other words, to assess Q1, Q2 differences, and also to assess the impact of deviations in the Q3 attributes. These attributes may be differences in apparent viscosity, differences in the global or particle size distribution, or differences in the formulation pH. I'm just naming a few here, between the reference and the test, and between the reference standard and the test product. Finally, modeling and simulation approaches are complementing in vitro characterization-based PE approaches, such as ones that include in vitro permeation testing studies and other methodologies for certain challenging drug products. All these applications are founded on the fact that the developed models are informed by drug product quality attributes that are specific for the reference and the test products, and that makes them relevant and powerful. Thank you.
Thank you for responding to that question. We do have one more question that just came in for Dr. Eleftheria Sakalazou, and here is the question. If an applicant is planning on using modeling and simulation approaches to support their alternative BE approaches, how can they receive the agency's preliminary feedback on the feasibility and implementation of their approach?
Thank you for this question. I have included the answer in one of my last slides, but I will take this opportunity to reiterate that we at the agency acknowledge the challenges associated with applying modeling and simulation approaches when supporting an ANDA. For this reason, we encourage prospective applicants to seek the agency's feedback on their modeling and simulation approaches through the pre-NDA meeting request program, as well as any other venue they may consider appropriate. We support modeling and simulation as part of an alternative bioequivalence approach, and we will be happy to provide guidance on the technical aspects of the modeling and simulation approach, as well as discuss the application and the role of an in silico model within the scope of a prospective ANDA. Thank you.
Thank you for responding to that question. We just have 2 minutes left in our Q&A panel, and we'll try to squeeze one more question in. And we just had a question come in for Dr. Feng Wu, and here is the question. What are the consideration points for expanding biowaivers to non-Q1, Q2, BCS Class 3 drug products?
Thank you for the question. For BCS Class 3 drug products, whether excipients would impact permeation would be an important consideration factor. As I presented, we have GDUFA-funded research to test the impact of commonly used excipients on permeation of BCS Class 3 drugs. As shown in the presentation, within a certain range, most of the tested excipients may not have significant impact on the permeation of the tested BCS Class 3 drugs. For which the systemic exposure is less likely to be affected. These outcome of these research studies may serve as the scientific basis for Biowaiver for non-Q1, Q2 BCS Class 3 drug products. Also, the in vitro permeation testing and the PBPK modeling may be used to support expansion of Biowaiver to non-Q1, Q2 BCS Class 3 drug products. Thank you.
Well, that's all the time we have for questions. I want to give a huge thank you to our panelists for answering numerous questions that came in once again.